Reproduction is the birth of new structures, but it occurs much more often at the cellular level. The purpose of cell division is for cell reproduction, growth, and the replacement of lost or damaged cells. Chromosomes are made up of a material called chromatin, a combination of DNA and protein molecules. The protein molecules help organize the chromatin and help control the activity of its genes. Nucleosomes consist of DNA wound around histone molecules, which are protein molecules. The cell cycle is an orderly sequence of events that extends from the time a cell first arises until it divides. The cell cycle consists of two distinct phases, interphase and the mitotic phase. The mitotic phase consists of 4 stages within itself: prophase, metaphase, telophase, and anaphase. Most of the cell cycle is spent in interphase. This is a time when a cell performs its normal functions within the organism. During interphase, a cell roughly doubles everything in its cytoplasm. It increases its supply of proteins, unincreases the number of many of its organelles, such as the mitochondria, ribosomes. The cell typically stays in interphase for at least 90% of the cell cycle. The most important event of interphase is chromosome duplication. The DNA in the nucleus is copied. This is the middle of interphase and is called the S phase for DNA synthesis. The period before S phase is G1. G stands for gapping. During G1, growth and normal metabolic reaction occur. The period after S phase is G2. G2 is the secondary growth phase. Growth and preparation for mitosis occurs during this phase. At this point, the cytoplasm contains 2 centrosomes. Within the nucleus, the chromosomes are duplicated, but they cannot be distinguished individually because they are still in the form of loosely packed chromatin fibers.
Prophase is the first phase of mitosis. During prophase, the nuclear envelope disintegrates. The nucleolus disappears, chromatin coils into chromosomes, centrosomes form mitotic spindles, and the centriole pairs duplicate, and spindle fibers attach to chromosomes.
The second phase of mitosis is metaphase. The meiotic spindle is fully formed and the chromosomes form on the equator of the cell. The centromeres of all of the chromosomes line up on this equator. For each of the chromosomes, the spindle microtubules attached to the 2 different sister chromatids pull towards the opposite poles. This tug of war keeps the chromosomes in the middle of the cell.
The third stage of mitosis is anaphase. During this stage, sister chromatids are pulled apart to the opposite poles and they become daughter chromosomes. Motor proteins at the centromeres move the daughter chromosomes along their microtubules toward opposite poles of the cell. This is when these microtubules that are not attached to chromosomes lengthen and push the poles further apart and elongate the cell. Some of the spindle fibers are short while others are long. This causes the cell to take on an oval shape.
Telophase is the final stage of mitosis. It is like prophase, but in reverse. Telophase first starts when the chromosomes reach the opposite cell poles. Then the nuclear envelope forms. The chromosomes uncoil to make chromatin, and the spindle disappears. Mitosis is now finished. Cytokinesis, or the division of the cytoplasm, usually occurs simultaneously with telophase. In animal cells, a cleavage furrow pinches the cells in 2, which produces 2 daughter cells. In a plant cell, a cell plate forms and begins to form a cell wall, which will separate the cell into 2.